Hi, everyone, and welcome back. In this video, I'm going to talk to you about the different human chromosomes that our cells possess and talk about their structure a little bit, what they look like, and the different parts of them, and a little bit about how that can impact the inheritance of the genetic material that is contained within them. So let's start quite simply with looking at what we see in the nuclear genome. So, human chromosomes, at least when we talk about the nuclear chromosomes, are they consist of chromatin, which is DNA wrapped around histone proteins. And as I've talked about in previous videos, they can either exist in a condensed state, a state that we refer to as heterochromatin, and a relaxed state, which is called euchromatin. And we talk about the ability of cells to change the structure of their chromosomes, to either relax them or to condense them, depending on what's going on in the cell. This is covered in my video on epigenetics. It really comes down to the modification of DNA and the histone proteins around which the DNA is wrapped. And this is a way of controlling gene expression. If the chromosomes are tightly wound up in heterochromatin, they're not really accessible to the transcription machinery. And in that relaxed euchromatin state, they are much more accessible, meaning gene transcription can actually occur. So when cytogeneticists are looking at chromosomes, and when you think about what a chromosome looks like, you are probably envisioning chromosomes that look something like this. So these are white blood cells that were trapped in metaphase. So they were just sort of grabbed out of metaphase, dropped onto a slide, which caused them to explode. And this releases all of their chromosomes. The reason why cytogeneticists do this is because in mid-metaphase, the chromosomes are more condensed than they'll ever be. They are as tightly wrapped up in heterochromatin as they possibly can be, and this makes it significantly easier for them to observe. And the reason why they're so tightly condensed like this is also makes it easier for the cell to move them about like they need to do during the process of mitosis. And in this condensed state, just to give you an idea of the size that we're looking at, the human chromosomes are about 5 micrometers long, and they're about 700 nanometers wide. When they are in their relaxed state, that same chromosome loosens up significantly to the point where they can be almost 2 millimeters long, and roughly 30 nanometers wide. So they get skinny because you're not looking at all these packed chromosomes. The DNA is not all packed in. It's all loosely stretched out. All right. What's interesting is it doesn't really matter whether they're in the condensed state or the relaxed state. All of that DNA still fits in the nucleus of every single one of your cells, which is kind of amazing. So when we look at the nuclear chromosomes, we are going to see two different types. First, you are going to find 22 pairs of autosomes. These are the chromosomes that we number 1 through 22. And then we also have one pair of allosomes, which are our sex chromosomes. So you can see here we're looking at a person who is a genetic male. And we know this because when we look at the allosomes, we see one X and one Y. If this were to be a genetic female, we would expect to see a pair of X chromosomes down here. Autosomes are actually numbered according to their size, so 1 through 22. And as you can see, as we go from 1 all the way down to 22, they get slightly smaller. So basically, 1 is the biggest chromosome and 2 is the smallest, and that's just how we've numbered them. And when we look at them this way, this type of arrangement is something that we call a karyogram. We arrange them by size, and cytogeneticists like to look at them this way because this is the easiest way when you pair them side by side like this to see if there are any type of chromosomal abnormalities that may show up. When we write out the result of a karyogram, we write it as a karyotype. The karyotype is going to involve a few key pieces of information. The first is the total number of chromosomes detected, which in a normal human is going to be 46. We also indicate genetic sex using the XY, which would make this a genetic male. So, for example, 46XY would make this a genetic male with no abnormalities. However, if there are abnormalities, we also have to annotate those as well. So, for example, if we see something that a karyotype listed as 46XX inverse 6 or INV6, this is a genetic female who has an inversion on chromosome number 9. Turns out that's actually fairly common. Turns out that doesn't actually lead to really any significant changes physiologically. Or you can get something more profound, 47XX plus 21. So this is an individual who has 47 chromosomes. This individual has an extra chromosome 21, hence the plus 21. They're also a genetic female. So in this case, 47XX plus 21 would be a genetic female with Down syndrome. All right. So that's how we write out karyotypes. This is the way cytogeneticists make annotations if they detect any abnormalities when they look at a karyogram. And this is the way we can convey that information. So another question we can ask ourselves is exactly how much DNA is in a cell. So geneticists measure this in two different ways. One of them is through something called the C value. C values are usually annotated in millions of bases. And the C value is set at the amount of genetic content in the gamete of an organism. So for example, in humans, the human C value is about 3,000 million bases, also known as 3 billion bases, or in science terms, 3,000 megabases. So these 3,000 megabases make up the genomic content of a haploid gamete, all right? An egg or a sperm in human beings. Now, one thing to note, when we look at a regular cell, a diploid cell, so a somatic cell, not a sperm or an egg, they're a 2C cell, right? They are going to have 6,000 megabases because they are diploid. But if you take that same diploid cell, you allow it to go through S phase prior to mitosis, you will note that they are now 4C. They now contain 12,000 megabases of genetic information. And this has become because during S phase, all of the chromosomes within that cell have undergone DNA replication. So you go from having 23 pairs of chromosomes to having 23 pairs of chromosomes, essentially. So we end up at 4C. So during the cell cycle, depending on which stage of the cell cycle you're in, which cell type you're in, a cell in a human can either be 1C, 2C, or 4C. Note, there is no 3C because there are no human cells that would have 3 times that number because it just wouldn't work that way. The other way we often look at the genetic content of a cell is what we call the N number. So the N number is the number of chromosomes in a gamete. So for humans, and this is probably something you're more comfortable with, for humans, our haploid number, 1N, is 23. There are 23 chromosomes in any sperm or egg cell that you find in humans, typically. Of course, there's always abnormalities. Then in a regular cell, if you look at a diploid, a somatic body cell, a cell that would undergo mitosis, you would say that cell is 2N. It's diploid. It has the diploid number, which is 46. But here's where things get a little wonky. When we look at a regular cell after S phase, you will note that our C value quadrupled. We're now at 4C, right? Because we've doubled all of the chromosomes. We now have 12,000 megabases of DNA in our cells. But the N number doesn't change. It stays right at 2N. And here's why. When chromosomes undergo doubling through S phase during the cell cycle, the actual chromosome number doesn't change. And the reason why is because the chromosomes that have just been replicated are all identical to another chromosome that already exists, right? So that's dad's chromosome 1 making a copy of itself, that sister chromatid, or mom's chromosome 3 making a second copy of itself to make that sister chromatid. Those don't count actually as distinct chromosomes, genetically speaking, because they contain the exact same information as another chromosome does. So as far as the cell's concerned, there are still really only 46 chromosomes in there. Even though they've all been duplicated, the genetic information is identical to a cell with 46 chromosomes. So while the C value continues to increase after replication, technically speaking, the N value does not. So that's two different ways that we can assess genetic information within a cell. And also, it's an important note in terms of how we assign sort of the value of an individual chromosome, right? C value versus N value. So as I mentioned, during S phase, we're going to get replication of our chromosomes. So another thing I just kind of wanted to remind you all of, and this is mentioned in my videos on mitosis and meiosis, and it's mentioned elsewhere. But it's just another thing that is often challenging for people to understand. When we look at a diploid cell, each autosome has a homolog, right? So homologous chromosomes are the maternal and paternal copies of a chromosome, right? So mom's chromosome 1, dad's chromosome 1. No, they will all have the same genes located in the same places along those chromosomes, but the information contained within those genes may vary, right? So because we have different alleles. So some of the information may be identical, other parts might be different, but they have genes located at the same locus, which is where the term homologous comes from. We also have non-homologous chromosomes. So those are two different chromosomes within the same organism or cell. So for example, dad's chromosome 1 and mom's chromosome 3, or even dad's chromosome 1 and dad's chromosome 3, those are non-homologous chromosomes. They do not contain similar genetic information. And this is where people get confused down here is with the sister chromatids. So sister chromatids are a replicated version of a chromosome. So if you take dad's chromosome 1, put it through S phase, and now you have two copies of dad's chromosome 1 right here, these two are sister chromatids. Here's dad's chromosome 1. It went through DNA replication. Now we have two. They're attached here at the centromere. Those are your sister chromatids. There's mom's copy of chromosome 1, goes through DNA replication. And now we have two copies of that attached at the centromere, and those two are sister chromatids. Together, they are referred to as homologous chromosomes or homologs, however you want to refer to them. All right. During mitosis, these sister chromatids will separate during metaphase. So that's where you get, all 46 duplicated chromosomes line up on the metaphase plate. Half go one way, half go the other. And because this process works this way, you end up with two identical diploid daughter cells. However, during meiosis, you're going to have one non-sister chromatid interact with the other non-sister chromatid through crossing over. That's during prophase 1. So that'll be one part or multiple parts of mom's chromosome 1 interacting with one of dad's copies of chromosome 1 in a sister chromatid. That's crossing over. That happens during prophase 1. That's one way we can introduce genetic variability through the process of meiosis. And then you also have the fact that these two homologous chromosomes are going to form a tetrad, and then they're going to separate through random assortment. That's also going to jumble up the genetic information as well. So it's important to understand these terms because we need to know what we're talking about when we're talking about meiosis and sort of the mixing up of genetic information. So if we look at one of our chromosomes and we kind of annotate what actually exists on each of our chromosomes, we'll find there are a couple of several key structures that we're going to find there. First and foremost, when you think about chromosomes, we're talking about DNA, so you're probably going to be thinking there are genes there. And you're right, there are genes all over the chromosome. So here they're showing them kind of interspersed over this little section. That's not true. Genes would be dispersed all over this entire chromosome. There are thousands dispersed across each of the chromosomes. Obviously, the larger the chromosome is, the more genes there will be. So chromosome 1, our biggest one, has just over 4,700 genes. But when you look at some of our smaller ones, like 21, 22, 23, they don't have near that many, but they're scattered throughout the length of the chromosome. You'll also find origins distributed about every 40,000 kilobases. So that's 40,000 base pairs. You'll find origins. Those are the sites of DNA replication. So, one of the things I mentioned during DNA replication in my videos, prokaryotes, they have a single origin because they have a circular chromosome and it goes all the way around. But with these linear chromosomes that are much bigger, they have ends. There's no going all the way around. They have origins scattered throughout the chromosome to make this process simpler and faster. In the, and I'm going to air quotes, middle of the chromosome, you are going to find the centromere. This is a region where no genes exist. It's just a simple repetitive sequence. And it's where proteins are going to attach during the mitotic process or meiosis, theoretically. So there really isn't important information stored there because it's really just an attachment site. Now, I say the middle with air quotes because we can actually describe chromosomes based on where the centromere is located. So many chromosomes are metacentric, and then we refer to them as having a slightly longer arm, Q arm, or a slightly shorter arm, the P arm. and we say it's roughly in the middle. You can also have some chromosomes in humans that are submetacentric. You can see this one's slightly off center. It's not right in the middle. So the P arm is much shorter than the Q arm in these submetacentric chromosomes. And then we also have some that are acrocentric, which means they are incredibly off center. This one's just slightly off. This one is, you can see the P arm is miles shorter than the Q arm on these acrocentric chromosomes. And then in some species, we don't find these in humans. We have telocentric centromeres that are located literally at the end. They just kind of pin them together like clothespins. They look like this. We don't find these in humans, by the way. And speaking of telo, that should remind you that our chromosomes are also going to have telomeres. So these are these repetitive sequences that cap the ends of our linear chromosomes. These are there to help protect our chromosomes from degradation. So if you recall, the ends of linear chromosomes slowly shrink after each round of replication because there's no place to put the lagging strand primer. So these telomeres are this repetitive sequence that helps to keep useful information from being chewed up at the ends of these chromosomes. There is an enzyme that can restore telomeres. It's called telomerase, but we don't find it active in normal cells with the exception of our stem cells. However, it's also been found to be active in cancer cells, which helps to protect them from the damage that would be associated with their rapid division. Now, the other type of DNA that we're going to find in our cells is in the form of mitochondrial DNA. So if you recall, mitochondria, as well as chloroplasts, which we don't have because we're not photosynthetic, are organelles that are believed to be of endosymbiotic origin, which means they are actually prokaryotes that were at some point in our evolutionary history absorbed by eukaryotic cells to perform functions. Essentially, mitochondria are the powerhouse of the cell. I'll just say it's literally a meme at this point. Mitochondria are the powerhouse of the cell. They produce the bulk of the energy through respiration. You also have chloroplasts, which are responsible for the photosynthetic properties of plants. So, and other photosynthetic organisms like Euglena, which is not a plant. But these still retain some of their own DNA. So when we look at mitochondrial DNA, we're going to note some key features of it. It does not contain chromatin. It is circular. It has a single origin. There is no mitosis or meiosis. So when you look at mitochondria, they simply divide through binary fission. Like, one interesting thing about mitochondria and chloroplasts, cells can't make more of them. They only arise through the division of an existing mitochondrion or existing chloroplast. So, and they do that through binary fission. There are also no centromeres or telomeres because it is a circular chromosome. And if we actually look at what's encoded within the human mitochondrial DNA, it's not much. So like I said, the C number, so the haploid number for our cells is right around 3,000 megabases, 3 billion bases. Mitochondrial DNA, 16.6 kilobases. So that's 16,600 bases. Significantly smaller. And that's because over the past 1.8 billion years or so since this has happened, the majority of the mitochondrial genes have gone to the nucleus through a process called horizontal gene transfer. And all that's left are about 37 genes. 13 of these encode mitochondrial proteins. They're almost exclusively involved in the electron transport chain. And then you also have several tRNAs and rRNAs. And one of the things we also have to acknowledge is that mitochondria have their own ribosomes. Oh, and by the way, those ribosomes are prokaryotic in nature. So all of these things act as evidence in support of the endosymbiotic theory, but it's also something that we have to reckon with because some very important processes that occur within our cell are dependent upon genes that are encoded within this mitochondrial genome. Now, each mitochondrion within your cell contains somewhere between 2 and 10 copies of that mitochondrial DNA, and it just gets partitioned when mitochondria divide. One of the things that people don't fully understand about mitochondria, like this drawing of a mitochondrion is probably very comfortable for you. It would probably be even more comfortable if it was like pink or purple, because that's how they're always drawn for some reason. But what people don't understand is that mitochondria are not static organelles. They fuse with each other. They divide all the time. The mitochondrial network changes on a regular basis depending on the physiologic state of the cell. I know a lot about this. I did this as part of my research, so I can go down this rabbit hole. I'm going to try to avoid it. But what you have to understand is that Mitochondria are constantly like dividing and fusing together. And what ends up happening is this mitochondrial DNA gets shared a lot. One of the things that's really interesting about mitochondrial DNA is it, because it's prokaryotic, it's not quite as good at fending off mutations that happen during the replication process. They're also the site of reactive oxygen species production, which means they're also that DNA is also subject to significantly more exposure to harmful products that can damage it. And as a result, the mitochondrial genome can get mutated fairly readily. Now, mitochondria typically handle this by if you're a mitochondrion with bad DNA, you're also a poorly functioning mitochondria, which means the cell selects you to get rid of you and your bad DNA. But if some of these mutations persist, they get shared with future mitochondria, and you end up with a really interesting phenotype for any sort of disease associated with this, because cells can be different in terms of their functionality depending on how much of the DNA in their mitochondria is mutated. So you can have some cells that function perfectly fine because they contain very little of that mutated DNA, where other cells are in rough shape because they have more of that mutated DNA. And this means when you look at mitochondrial diseases, they often vary wildly in terms of their presentation, their severity, and what tissues are impacted. So kind of a little tangent, but something that's really neat to understand about mitochondria and mitochondrial related diseases. The other thing that's super duper interesting is that mitochondrial DNA are always inherited from the mother. It always comes from the egg. So one of the ways that we can trace human ancestry back to its origins is actually just looking at mitochondrial DNA. And we can actually see how related so much of the human population is by tracing our mitochondrial DNA back. And we'll find that there's actually not a huge number of what we would call mitochondrial eaves, where these mitochondrial DNA sort of dead end the origin of that mitochondrial DNA. We also have the sex chromosomes. So these are, in fact, nuclear chromosomes, but because of the way they're inherited, they can behave differently in terms of inheritance. So when we talk about sex determination through chromosomes, the first thing we're going to have to figure out is what species are we talking about. So if it's a mammal, a fruit fly, some dioecious plants, then we are talking about a sex determination system that involves X and Y chromosomes. In these species, having two X chromosomes makes you a genetic female. Having an X and a Y makes you a genetic male. We also have an entirely different system when it comes to birds, moths, and butterflies. So they use the ZW system. And weirdly, in this case, if you have two Z chromosomes, you're a male. All right. So in this case, the homogametic individual is the male, whereas in mammals, fruit flies, and plants, it's the female. When I say homogametic, I mean they have two X's or two Z's. On the other hand, the females are going to have one Z and one W chromosome. And this is just two systems that we're going to talk about. There are others. For example, in nematodes, if you get two X's, you're a fully functional hermaphrodite. And if you get one X, you're a male. Males are XO. They get one sex chromosome, but no other. That makes them a male. And then if you have two X's, you're a hermaphrodite, which means you actually produce sperm and eggs and can fertilize your own self. So species vary wildly. And one of the reasons we know this is because one of the reasons this has happened is because sex chromosomes have evolved several different times. This is one of those things where, like, we're looking at, if you want to go back to evolution, these are examples of analogous structures, right? These sex chromosomes, they do the same thing, but they're structured differently. They behave differently. And then again, if we're looking at individuals that are XY or ZW, we would call those heterogametic. So one of the things that has to happen, right, is we get meiosis and we get these pairings and we get this crossing over and all that kind of stuff. So during meiosis, if you are homogametic, your sex chromosomes will just pair like normal. They're going to act like autosomes. Like it's very simple to understand because you have two X chromosomes, you have two Z chromosomes. However, if you're heterogametic, it gets a little weird. The sex chromosomes are going to pair with each other during meiosis. And then also the one thing we have to acknowledge is that even though the X and Y chromosomes are very different in terms of their structure and the genes they contain, they also have what are called pseudo-autosomal regions. These are located mainly at the ends of the chromosomes, but also near the centromeres. And this actually does allow for some pairing to occur. And this also allows for crossing over to occur within these regions. Interestingly, in these regions, there are some shared genes. So, genes that are in the pseudo-autosomal regions of X and Y chromosomes will actually be inherited in a Mendelian fashion. However, if it's something that's in the X specific or the Y specific region of those chromosomes, they're going to be inherited in a manner referred to as sex linkage. So sex-linked traits are going to deviate from classical Mendelian patterns of inheritance. And we are talking mainly about genes that are found on the X or the Y chromosome or the Z or W chromosome, depending on the organisms, but not found on the other, right? So things to know, I'll use X as an example. Father's X-linked genes are always passed to his daughters, right? Because during the process of forming those gametes, the father can either give an X or a Y to the resulting gamete, to the sperm cell, and he's going to give one or the other. In order to make a genetic female daughter, he would have to give his X, because if he gave a Y, it would result in a genetically male child, son in that case. So dad's X-linked genes are always going to the daughter, whereas mom's X-linked alleles are going to be shared evenly between sons and daughters, because moms can only give an X, right? And they have 2 copies of it. So one interesting thing that we can do to determine if a trait is sex-linked is to perform a reciprocal cross. So let's talk about what this looks like. So if you recall from Mendel's work, one of the things he did with his pea plants was what we now refer to as a reciprocal cross. He pollinated female plants of one color with pollen from a pea plant that has a different flower color, and then he reversed it, right? So for example, he might use pollen from a purple flowering pea plant to pollinate a white flowering pea plant, or he can use a white flowering pea plant to fertilize a purple flowering pea plant. He got the same results, which tells you the traits that they were looking at were likely not sex-linked. If you do a reciprocal cross and you get different results, that would likely indicate it depends on the sex of the parent, which means this would be a sex-linked trait. So let's look at an example of how we can do this using the XY system in Drosophila, fruit flies. There is a gene in Drosophila that controls eye color. It is referred to as W. W stands for white. Now, I need to apologize for geneticists. The behavior of geneticists is such that we name genes not for what they do, but for what the appearance of the phenotype is when we break it. So when we break this gene, we mutate it, the eyes turn white. So we call the gene white, even though the function of that gene product is to turn the eyes yellow. And they do this, I believe, just to confuse everybody during their first years of graduate school. I'm just kidding. But it does serve the function of confusing everybody who studies this stuff, because we're basing it off of the phenotype that appears when you mutate it, not the phenotype that is there when it's not mutated. So anyways, fruit flies who have a W plus version of the gene, the wild type version, have red eyes. All right. It is a homozygous dominant allele. So even having a single copy of it will produce red eyes. So here we have 3 different females, homozygous dominant or homozygous for the wild type, red eyes, heterozygous for the wild type and the white mutant, still red eyes, homozygous for the white mutant, now they have white eyes. But if you look at males, they are XY. So if a male has a single functioning wild type copy of the white gene, they will have red eyes. And if they have the mutated version, they have white eyes. They have no compensation. They cannot be heterozygous for this particular allele. But let's say we didn't know that. Let's say that we didn't know that because we don't know that this is a sex-linked trait. How could we perform a genetic cross or a set of crosses that would help us determine that? Well, it's simple. You set up 2 different parental crosses. First, you use a homozygous wild type female with red eyes, and you use a mutant white-eyed male, and you mate them together. Do the same over here, except it's the reciprocal. Now you're dealing with the homozygous white-eyed mutant versus the wild-type red-eyed male. When you go to the next generation, you're already going to start to see the differences. When you go to the F1 and you look at the F1 generation, you're going to see that the individuals that result from this are a mixed bag. Here, because the female is going to pass on its white-eyed the wild type white gene onto all of its offspring, male or female, they will all have red eyes. Because no matter who you are, you have a wild type copy of that white gene to give you your red eyes. But down here, the female can only give the mutant alleles. So while her female offspring are going to be rescued by dad's wild type white allele and have red eyes, there's no compensation in the resulting male progeny. And you can see all of a sudden now you have white-eyed males. So when you get to the F2 round of this cross, you're going to start to see other differences. So when you allow these F1s to mate with each other, control the mating, you will have red-eyed females and red-eyed males from the first cross. And the resulting progeny, you're going to see this. You're going to have red-eyed females, you'll have red-eyed males, you'll have more red-eyed females, and some white-eyed males, right? Here are homozygous wild type. We have our mutant males, and then the rest are either a wild type male or a heterozygous female, which appears red-eyed. So you have a 3-to-1 phenotypic ratio here. However, when you look at it over here, now you're going to have 50-50. Red-eyed females, red-eyed males, white-eyed females, white-eyed males. This is the major difference. And this is how sex-linked genes reveal themselves when we do genetic crosses. You don't get the standard pattern of Mendelian inheritance when you do the reciprocal crosses. You don't get identical reciprocal crosses. If this were a gene on a somatic chromosome, not one of the sex chromosomes, you would actually be able to see that they would be inherited in a Mendelian fashion. You'd probably end up with this 3-to-1 ratio no matter which parent you went from. Now, you're probably thinking, how do we do this in humans and other animals where we can't just set up genetic crosses where either time would be a constraint or just ethics would be a constraint? Well, this is where pedigrees come from. So I'll be doing another video soon on pedigrees. And pedigrees, we can go back through, and while they're not quite as accurate as controlling crosses, we can start to look at the inheritance pattern of certain diseases or mutations or inherited traits, and we can see, is there a sex-linked based pattern on this? And I'll talk more about that in that video. So just keep that in mind. We also have the case of Y chromosome linkage. So the Y chromosome does not contain a lot of genes. It's most of the genes have been transferred over to the X chromosome, except those in those pseudo autosomal regions that we talked about. So when we look at Y chromosomes, when we look at Y-based linkage, these are traits that are only going to be inherited by males. These are not genes that we find on the X chromosome at all. One particular trait that we find in humans is something called auricular hypertrichosis, which is long hairs growing from the rim of the ear. This is something that is passed around in certain cultures, and it does show Y-linked inheritance. We see a phenotype linked to feather color in turkeys. This is a Z-linked trait. The one thing to note here is, remember, in birds, it's the females who are ZW, which means we typically see Z-linked traits appear much more significantly in female birds, whereas we see Y-linked traits appear predominantly in male humans and other mammals. So that kind of wraps up the conversation about the structures of our different chromosomes, helps us understand why they look the way they do, why the things that they contain are inherited in the way that they do. So I hope you learned a lot. I look forward to seeing you again. Talk to you soon.